Welcome to Study Time, a televised home learning program produced by Rwanda Basic Education Board.
Welcome viewers once again. Here we are for the TV series for lessons that are going on. As we had talked about before, we are still in Unit 5 when it comes to the Senior 5 class. And today we are kind of wrapping it up, if I would use the words, in terms of the last component of meiotic cell division, in which we are to look at what we should take as a package back home. So, what do we have? We should be in position to describe how we vary, how other organisms vary, because of what happens in gamete formation. So during the time when gametes are forming, how does meiosis bring about a difference? What is the actual cause? Remember, the other time we looked at the process going on, meiosis as a process going on, the 3D activity, and also an overview of exactly what happens. But now this time there are two important things I want us to focus on. Exactly what happens when the chromosomes kind of switch content, and at the same time when they pair along the equator and then they separate. All these, what do they come up with? But also, what if the process of separation jams? So here we go, follow the laser. Non-disjunction, when does it occur? And if it does happen, what is its effect on the organisms that are going to be produced, the offspring? If they are good or bad, this time around we are focusing only on humans, where in most cases things don't turn out to be right. But do we appreciate some of these changes? When they do happen, what value do we attach them to? If there happens to be children that are born and we have some of these as kind of disabled, as we have always referred to, which is not right, does it mean that they are completely unable to do most of the things? Does disability equate to inability? That's where we have to strike that thin, that gray line, that gray picture. What is absolute truth and what is real truth? What is the relevance? And what is the relative nature of these two diversions? So, that's what we have to capture today. But it all starts here. If we are to have new organisms to form, magic has to happen. The miracles of life, reproduction, fertilization. Remember, I know most of you are looking at the picture and thinking, oh, wow, we are going to study reproduction, which is always an excitement to every learner in high school and secondary. But, guess what? It's not. So, cool down. We are looking at when meiosis occurs and fertilization has taken place, how do we come out different? So in the background, we have a gamete, male, and the bigger picture of an egg. Penetration, get in, have the nuclei fusing, and we have a zygote. So, when meiosis occurs only in males and only in females, gametes are produced. But these fuse, which we have as fertilization. Remember that we have infinite combinations of possibilities in which these chromosomes will arrange themselves, if you can remember some time back. So, the switching of the genes, which is so multiple, and resulting into increased gene variation, producing more resilient populations, in which we have a lot of differences among us. Who is prone to attacks by changes in the environment, who is able to resist, who has the toughest features, who has picked features from mom and features from dad, and they make a perfect combination that this child actually is able to survive much better than the parent. So if we look at that as differences and adaptation according to changes in the environment, we also need to think How do we evolve? Should be an interesting question. Yeah, how do we evolve? Is it because a lot of things have happened around us in the environment, and also based on the new features we have from our parents, completely different from one another? You differ from your other siblings. Oh, so who makes it to the next phase of life? Who survives this COVID? What does this person have? Some go through it, others don't make it out. What's the speciality? How does their metabolism fight off this low uptake, this attack attack onto the red blood cells, this attack into the respiratory tissues? And the same applies to other diseases. Remember, once a change has taken place and is actually happening in the gamete cells, like we said the other day, this is inheritable, so it's passed on. If it's an error, take over. If it's a better feature, then pass it on and get better adapted. But along the way, then we end up evolving. Over time, life changes in terms of form and metabolism, and generally, a new species arises. But where is the very beginning of all this? Which takes us to two important things that do happen in prophase 1, if you can remember, and also in meiosis 2. Prophase 1 in meiotic division 1, prophase 2 in meiotic division 2, and so on, like we said, phases in division. So in the process of the switching of the genes, like we said last time, your siblings at home, you switch shoes and walk away. But remember, the left to switch with the left and the right to switch with the right. So here we go. Look at the image. Two homologous chromosomes. So it's a pair. And they swap. Capital A here and small a to the right. What is the result? Switched, we separate them, and they have a mix. What is crossed here is a linkage group, which actually contains genetic material. Now, they have swapped information, they have separated. Now the question is, once they have reached these cells and we have the first meiotic division, what do we actually have? So try to recall what we did in the previous lesson, which I'm not going to repeat over and over again. New combinations. But in the second meiotic division, again, a division will occur here and a division will occur here. So we now have 4 of them. Like we said, we would actually find that each of these gametes is different from the other. They are going to fuse, like we said in the previous slide, and you have magic. Life resumes. A zygote is produced, continues. Different features because of the different genes. So to wrap it up, the purpose of this crossing over generally brings about combinations, giving you genes peculiar to yourself, completely different from your sibling, and as well your parents. So one part you say, uruokwande mama. Njendo kwa shangazi, shangazi na bwari zina. But the question is, says, yeah, I think I'm seeing the mom in her face, then I'm seeing the dad appearing in the ears and the cheeks, or maybe the way they walk, which takes us to the inheritance of the features that do appear, and so the difference. Some of these genetic differences, the diversities, the variety, resulting from meiosis, we have to benefit from that. So when we always talk about meiosis as a cause of variation in sexually reproducing organisms, we actually go slightly deep inside and pinpoint to those 2 events that do happen in meiosis: crossing over, random assortment. The paired chromosomes can now exchange segments, which we talked about as linkage groups of DNA, via a mechanism which we just talked about. So that is the purpose. Meaning, therefore, that when we look at generally what happens in life, we are not using humans here, so maybe you see mom and dad here and stuff like that. No, let's use fish. It's more humble. So we do have adults undergoing meiosis, producing haploid cells, egg and sperm. And these unite, so they fuse. Then a zygote is produced. In juvenile stage, the young stage, like you back home, toddlers, this is where you are. I'm not saying you're fish, get my word. Then you will grow until your parents say, but what are you talking about? Why don't you think you should be in your own house? So after having you all say that, what next? How do you stay alone in the house? Who is taking care of you? As if women are married to take care of men. That ends. If you still have that impression, you're finished. The purpose is pass on your genes to the next generation. One would literally say that once you have produced, you've actually died because your purpose is over. Pass on the information before you disappear. So once you're born, and it's okay, the parents can pass on. After all, you already have them in your body. So the cycle goes on and on and on. But something interesting here. If we had 2n and we now n, then we go back to 2n. It means that the purpose of meiosis is to maintain species. I don't know whether you get that. The purpose of meiosis is to maintain species. So we've remained humans for I don't know how many millions of years. Think about it. We've never out of nowhere after giving birth to a child and the child grows and then he is also giving birth and produces fish. No. Fish separates species, human race separates species. And we've stayed like that for a long period of time, way back. And we shall continue like this unless something unusual happens. So maintaining the chromosome number as a diploid state requires some form of meiosis to occur properly, so that we have to reduce to n. So, we also are eukaryotes, we reproduce by sexual reproduction, and all other organisms do go through the same. But a few of them are able to undergo asexual. What does this mean? Minus the use of gametes. It doesn't mean that they completely have no ability to produce gametes, but depending on environmental conditions. Say, for instance, if we talk about ubushishi bwabo buriya burya ibishimbo bakateramo imiti kugira ngo basarure neza. So exactly what is happening here is that in the fusion of gametes, the sex cells, it's when we actually must have our diploid state coming back. So it's a compensation. Fertilization is a compensation for loss here, so that we go back here. So preventing doubling of the chromosomes means that we must undergo meiosis, reduce the numbers, so that we maintain the same fish, young, old, young again. So how do we actually see these chromosomes? There is something we talked about some time back about the karyotype, the arrangement, the positioning, and the sizes of the chromosomes. Way back when we were talking about DNA structures and the chromosome and the description of the chromosome and how many are they. What is the difference between one chromosome and the other? If we have 46, how different are they? Are they all the same size, shape, content they carry as genes? Which takes us to what we look at as a cardiogram. An image, a photograph, a diagram showing the chromosomes present in the nucleus of a eukaryotic cell. The arrangement follows homologous composition, so we look at them in terms of, okay, pair them, size. So we have a pair, So if we count 23 up to here, then we multiply by 2 to have 46 chromosomes. If we were to look at this in terms of, I would actually say, if I'm to literally count, Then I have 23 pairs, then multiply by 2 to get my 2n, which gives me my human or our karyotype. But the question is, this is a little bit smaller than that. But the rest of the others kind of look of more or less same size in a way, meaning that maybe one for male and then female. Remember sex determination in your O-level, X and Y. The Y chromosome is a little bit shorter. The position of the centromere is different from the X. And so would be the height, and so the message they carry. Let's not focus so much on that. So, if that is what we expect from every cell, what exactly are we having here? When meiosis has taken place, right, we expect 4 haploid cells to form, each different from one another, and as well different from the parent. But can we have some mistakes happening? One of them would actually be if the process of separation here jams, that we may probably have some gametes forming as empty at a given stage, or more. So you have 1 chromosome less or 1 chromosome extra. So we refer to this as non-disjunction, when the chromosomes fail to separate at anaphase. It can be anaphase 1 or can be anaphase 2. And that's why in this sentence here, I've not clearly focused on 1. But the pictures are giving us, if it happens in meiosis 1, what exactly happens? If it happens in meiosis 2, what is the result? Look at the images very well. 1, 2, 1, 2, and then the separation. When we have these cells separating, we have a problem. There is a cell going to form here, which most likely is this. Check that very well. If I can get a pen and pick out the red and move where we here, can this take us to here? And or can this here be the one, let's pick another one, say this section here be the one responsible for this? Think about it. So that at the end of the day, we have 1, so it's going to be 23 plus 1, 23 plus 1, 23 minus 1, 23 minus 1, 23 plus 1, and what else do we have? A 23 minus 1, and then you have our normal ones here. So you can check the results. They're so variable that at the end of the day, the karyotype we would now get, the cardiogram we are going to get, would actually look like this. So when we talk of triploids, from haploid, then diploid, then triploids, this is what we are having. Check this out. Instead of a pair, we actually have a triplet, commonly known as Down syndrome. And the individual here possesses 3 chromosomes at position number 21, meaning that the total, if we were to count all together, when we reach here to reach 23, we add the 4th, and then they become 47 in an individual. Now, the question is, once this has happened, how do we relate this to what goes on in our lives? Think about it. What is the right age to marry? Interesting now. At what age are you now, if you're male or female? Have you got a child yet? So if we look at the figures here, we are looking at the risk of a child having trisomy, such as Down syndrome, actually increases greatly with older ages, when mothers are aging. What is the chance that their gametes are actually going to form and errors result? So you have two lines, trisomy, 21, you have the line here, and then all chromosomal abnormalities are here. Now let's check the age. Some of these errors start happening even at 20, past 20, before 30 years of age. But now the question is, what exactly is happening? Is it 40, 41, 42, 60? Do we reach? But when you look at the data, it actually shows that some of these possibilities do happen even well before we actually hit our menopause. Should it be a worry? And if it is, how are you going to know that you actually have? Which takes us to methods that are used to obtain cells for karyotype analysis. So you know, once you're pregnant, what is the chance that you're going to have such a possibility? So, and here we do have ultrasound. You've all gone to the hospital, escorting your mom, or maybe you out there. One time they will have to check what is happening inside your baby as you're expecting the baby. But based on the age you have, or if you want to check exactly what happens as your baby grows, are there any abnormalities you need to prepare for before it actually happens? Is it a possibility to give you a chance to think about abortion? Think about it. So as the cell develops, as the zygote develops into an embryo, the cells form layers, so the core, the mantle, where all body organs are forming. And then the chorion and the amnion, all those components that do form surrounding the baby can also have traces of these cells from the embryo, from where we can extract them and map out our cardiogram. Then we're able to tell that, yes, I'm expecting, but the chances are that I might come up with this. Which takes us to this circle here and the 4 pictures we are having here. Question is, do you really need to think of abortion? Because you know that from the results during pregnancy, a possibility of trisomy is resulting. They are all children. Look at the way they smile. There's nothing wrong with them. So from the cardiogram, we are able to check where the error is, and then we are able to prepare both emotionally and all the physical and components requiring us to prepare for the child that is yet to come. If you're able to prepare for a baby girl because you've gone for a sex search, which we normally refer to as a penis search, what about this? In which way are you going to prepare? When the child comes up, are you going to think about preparations? So, let's check what we essentially would always develop some bit of curiosity to start thinking. I happen to have taught one of the classes way back, I think between 2005 and 2008. One of the kids in my class was actually a member of this category. And guess what? In every other person, there is always a diamond. You just have to mine it. And it's bright and it's shiny. It glitters. You know what I'm talking about. They ain't any different from you, and in most cases, they're actually better than us. But what do we need to be on the lookout for? The physical features that we think that are an indication that something like that would actually happen. Listen, this is a word of caution. Whoever is out there listening, I don't want you to start looking at your brother's face, or sister's face, or cousin, or mom, or dad. But you're looking out for all these features I've posted. Start looking at their palms and say, how many lines do you have in your hands? What about the size of your ears? And what about the eyes? What about the size of your neck? No. be positive, be objective, and consider everyone equal. That's the take home, the major important part of this component of the lesson. You can't be that mean, I know. You're good, you're well brought up, your parents gave you the right education, and I'm giving you a package. Please, be very considerate of this. Remember, like I said at the end, that we shall also have to think that in every person you come across, there is always something so special about them. Look at them. These are a few examples I've been able to come across. Actually, this image is extracted from the Center for Disease Control. There is always a talent. There is always a very strong ability in every individual. They can be able to do awesome things. So before you get these results and start thinking, oh my God, this is what I'm expecting once I give birth. How do I even prepare for this? Do I have the money to take this child to the right school that is purely inclusive? Thanks to the government of Rwanda, it has included this in our education system. All learners, inclusiveness. I love it. But I still remember one thing. Among the children I taught, between 2005 and 2008, I wouldn't have to mention the school, there's that one child who was always coming out. So caring, considerate of everyone in class. And I just asked myself, what exactly does happen for this to be like this? Why do we think that people we think that are actually kind of unable, they are actually more and very able than we really think? So what do we do? Drill and look for that diamond. You'll get it, and you'll love it, and you'll never let go. She was my best company in the whole class, if I would mention the words, to put it the list. So, yeah, with that, we come to the end of meiosis and its mistakes and how we can benefit from them, as well as taking a positive note out of it that even when these things happen, we still remain children. We need the very attention others are given because we can as well shine like that diamond. So with that, thank you so much for today and hope to see you soon.